Gastric secretions are regulated in two ways or by two phases, the cephalic phase and the gastric phase. The cephalic phase of gastric secretions occurs when sensory structures in the head respond to food-related stimuli. So it is sight of food, taste of food, or smell of food. So all three of these will trigger an increase in the amount of acids and the amount of digestive enzymes that are produced by the stomach. The other phase is the gastric phase, which is the longest phase of gastric secretions, which lasts about 3 hours, which is about 1 hour short of the typical amount of time that food spends in the stomach, as food typically spends about 4 hours digesting in the stomach. And this is the period in which this is the phase in which most of the gastric secretions are released. It's activated by stretch of the stomach, so that is, it's activated when food actually begins to fill the stomach. And it's inhibited by low pH as acids build up between meals, because what will happen is that as food is digested and moved into the small intestine, acids in the stomach will continue to be secreted, and the pH will continue to get lower and lower. That is, it will continue to become more acidic. And so that will influence a negative feedback mechanism to slow down the release of hydrochloric acid or the production of hydrochloric acid, as well as slow down the release of gastric enzymes. So in the stomach, particularly from the perspective of the smooth muscles, the three layers of smooth muscles that make up the muscularis externa of the stomach, the muscles contract in a way that essentially pushes the food kind of in a wave-like motion towards the duodenum. That is, the muscles will contract, pushing the food towards the pylorus. So remember, the pylorus is the funnel-shaped region of the stomach that leads to the duodenum. And so what we see is that we see in the propulsive phase peristaltic waves, that is, regular waves of contraction and relaxation, will push food from the body or from the fundus through the body of the stomach and to the pylorus on its way to the duodenum. There is also some grinding and mixing action that occurs at the pylorus. So this also helps to further break down the chyme mechanically, as well as regulate or at least provide the muscular action to push some of the food through the pyloric sphincter into the duodenum there. And so the pylorus itself is really kind of behaving as a pump that's delivering small volumes of chyme into the duodenum. And then the duodenum itself is regulating how much chyme is entering it. So as I mentioned earlier, the stomach usually empties itself in about 4 hours, depending on the size of the meal. If there is a lot of fatty chyme present, so that is, if there is a lot of lipids in the food that you've ingested that's needs to be digested, the presence of fatty chyme increases the amount of food and the amount of time that food spends in the stomach by about 2 hours. So the stomach takes about 6 hours or so to empty when fatty chyme is available. When sugary chyme is available, on the other hand, that actually decreases stomach emptying time, so the stomach is emptied much sooner than 4 hours on average. Because carbohydrates aren't really broken down very much in the stomach. Remember that the stomach is mostly the site of protein and lipid digestion, but we don't really get a lot of carbohydrate digestion going on in the stomach. So generally, carbohydrates don't spend a long time in the stomach, but proteins and lipids will. So this feedback loop that we see here just kind of highlights the fact that the presence of fatty chyme or acidic chyme in the stomach will slow down the rate of stomach emptying. And so that also influences the smooth muscles that are controlling the movement of chyme into the duodenum. And so that will decrease the contractile force of the three layers of the muscularis externa of the stomach, which will also decrease the rate of emptying in the stomach. So that way, fatty chyme or acidic chyme will spend more time in the stomach so that it has a much more sufficient period of time in which to digest. Right? Looking at some homeostatic imbalances, emesis is the term that we use for vomiting. And typically vomiting results as a fact of, as a result of extreme stretch of the stomach. So if we consume too much, and there's a lot of material in the stomach, that can trigger emesis. And also intestinal irritants like bacterial toxins, alcohol, spicy foods, things like that can be a trigger for emesis or vomiting as well. One of the things that we see with excessive vomiting, or at least overly excessive vomiting, not only does it lead to dehydration, we saw that the stomach uses about 3 liters of fluid, or it produces about 3 liters of gastric juice a day. And most of that is going to be water. And so excessive vomiting is going to cause pretty significant dehydration because we're going to be losing a lot of water. It also causes electrolyte imbalances. And it also results in particularly high blood pH. So what will happen during vomiting is we lose stomach acid, and our stomach will try to replace that lost acid by creating new acid. And so what's going to happen, remember that process of creating hydrochloric acid involved CO2, and it involved converting that CO2 into carbonic acid. And then we took a hydrogen ion off of that carbonic acid, and what remained was bicarbonate, and that bicarbonate was pumped into the stomach through the alkaline tide. And so when our body is trying to replace a significant amount of that stomach acid that was lost, it's going to put a lot of extra bicarbonate into the bloodstream, and so that will raise our blood pH. Our blood pH will become slightly more alkaline as a result of that massive influx of bicarbonate into the bloodstream. So that does it for the stomach. And before we look at the structure function of the small intestine, I'd like to take a moment and kind of divert to looking at some of the accessory organs that are going to be involved in secreting things into the small intestine. So here we'll take a look at the liver and the gallbladder. Now the liver serves a couple of different functions for our body. The main function that we think of for the liver is not a digestive function, but it's a cleansing function. And so we see that the liver has a really extensive network of capillaries through the hepatic portal system that delivers that blood coming from the digestive system to huge populations of cells in the liver that function to cleanse the blood before it re-enters the systemic bloodstream and goes back to the heart. So the liver has a huge function in cleansing the blood that's leaving the alimentary canal. As far as its digestive functions are concerned, the liver is one of the largest glands in the body, and its glandular functions include producing a substance called bile. And what bile does is that it doesn't digest lipids chemically, but it digests lipids or breaks down lipids mechanically. So bile emulsifies lipids. So it basically acts like a detergent to break up lipids into much smaller droplets or much smaller pieces. So that way the lipases and the lipid breaking down enzymes can have a much easier time digesting that lipid in a smaller period of time. And so it helps to really help complete the process of lipid digestion. So bile breaks down lipids by emulsification. So it's a mechanical process that bile breaks down lipids rather than an enzymatic or chemical process. The liver has a couple of lobes associated with it, 4 lobes in particular, none of which will actually specifically identify on your lab list or anything like that, but it is a lobed organ. So we'll look at a couple of the ducts that exit the liver and drain as secretions into the small intestine. The main duct that exits the liver is the common hepatic duct. And so there's lots of smaller ducts from various aspects of the liver that actually merge into this common hepatic duct on its way out of the liver. We also have the cystic duct, which connects to the common hepatic duct, or at least connects at the intersection of the common hepatic duct. So the cystic duct serves to connect the gallbladder, which is located here, to the common hepatic duct. And at the point that the cystic duct and the common hepatic duct meet, that structure becomes or forms the bile duct. And so think of the bile duct as the junction between the common hepatic duct and the cystic duct. We'll look at the gallbladder in just a moment here. The gallbladder's function is a storage site, and what it does is it stores bile that is produced by the liver. So the liver can secrete bile directly into the bile duct, or the gallbladder can contract and secrete some of its stored bile into that bile duct. The cleansing function of the liver takes place in the liver lobules. And the liver lobules are these regular blocks, if you will, or regular segments of the liver in which a central blood vessel is surrounded by the hepatocytes, that is, the cells that are cleansing the blood before it goes back into systemic circulation. And the lobules are organized in these hexagon-shaped structures. And so you've got a central blood vessel that we'll see in just a moment has lots of little capillaries that fan out from it so that those capillaries can deliver blood to the hepatocytes, and then the hepatocytes can cleanse that blood and send it back into the venous circulation. So we see that the liver lobules are the functional units of the liver, and they're actually cleansing and filtering the blood from the digestive system. A little bit closer look at the liver lobules. You can see the central vein in the center of the liver lobule, and it doesn't really look like it here, but all these little spaces are actually small capillaries that exist between the hepatocytes there. And then each of the lobules is separated from one another by a connective tissue septum, or a connective tissue barrier there. In diagrammatic form, we see that the central vein fans out into these very small sinusoid capillaries. So these are very leaky, very permeable capillaries that serve blood to the cells in the hepatic plates. So the hepatic plates are these sheets of hepatocytes that are adjacent to those liver sinusoids, or those small liver capillaries. So just to remind you that sinusoid capillaries, we reviewed these back in the blood vessels lecture here. Sinusoid capillaries are the most permeable and occur in only very limited locations. One of those happens to be the liver. So they have very large holes, or very large openings in them, so that fluids can leave the bloodstream very easily and enter those hepatocytes, where the hepatocytes will cleanse them. So we see that very large molecules and even some cells are allowed to pass through their walls. Red blood cells typically are too large to get through into the liver, so they don't plug up any of the vascularization of the liver. But these capillaries are very, very permeable with their large intracellular clefts, right? That is the large spaces between adjacent cells, large fenestrations, that is very large holes that exist within the cells, so that materials can very easily leave the bloodstream and enter the cells of the liver. So let's take a look at the hepatocytes for a second. And the hepatocytes are the cells in the liver that are doing the detoxification and the cleansing of the blood that's entering them. So the hepatocytes are specialized for cleansing because they have an abundant amount of smooth endoplasmic reticulum. Remember that this is the organelle that serves to detoxify things like drugs and alcohol. The hepatocytes also have large numbers of peroxisomes, and peroxisomes make hydrogen peroxide, which is H2O2, from oxygen and water. And so they use hydrogen peroxide to detoxify harmful substances that they find in the blood. And hydrogen peroxide is very toxic even to our own body that produces it. And so the liver hepatocytes actually produce an enzyme called catalase that serves to break down hydrogen peroxide very quickly. So that way, hydrogen peroxide is made, it's used in detoxifying, and then it's very quickly eliminated, so it's not hanging around. abundant Golgi apparatus, which helps to export materials from the cell. So we see that that's going to be really important in getting materials into the cell for the purposes of detoxifying. And then abundant mitochondria as well, because the hepatocytes are very energy dependent. So they're very dependent on ATP production so that they can continue functioning to cleanse the blood. Right. The hepatocytes are also producing bile because they are serving a glandular function. They produce not a ton of bile, about 900 milliliters or so just shy of a liter of bile per day. And a good amount of that is stored in the gallbladder itself to be released later on. So what is bile? Bile actually takes a salt form. So the hepatocytes produce bile, and then it's stored in the gallbladder as bile salts. They're a derivative of cholesterol, and as I mentioned, they emulsify fats and lipids. So that is, they mechanically break them down like a detergent so that they have smaller surface area, and it's easier for the digestive enzymes in the stomach and the small intestine to break down those lipids, right? And bile also contains a waste product from our blood, a waste product called bilirubin. And bilirubin is a leftover pigment from the breakdown of the heme group in hemoglobin. Remember, the heme group, part of the heme group contained the iron atom that binds oxygen, but the iron is typically reused to make new blood cells. We don't tend to lose a lot of iron through our waste or through the waste processes that are cleansing our body. But there is a leftover pigment called bilirubin that is eliminated. And this is injected into the small intestine with bile, or as part of bile, as a waste product. And when it gets to particularly the large intestine contains bacteria in it that break down bilirubin into a brown pigment that's called stercobilin, and stercobilin is the brown pigment that makes fecal material brown. This diagram here shows that the bile salts that are secreted from either the gallbladder or the liver are secreted into the duodenum, and we'll see the point at which they're secreted into the duodenum in a later lecture. Bile salts make their way through the intestinal system. They break down lipids by emulsifying them, and then about 95% of them, once they reach the end of the small intestine at a region that's called the ileum, they are reabsorbed into the bloodstream, and through the hepatic portal vein they're actually sent back to the liver, where they are stored and reused again. So the liver only has to make or manufacture a little bit of bile salt at a time because it can reuse some of the bile salts that it's made previously. So concluding with the hepatocytes here, we also see that hepatocytes function to, in addition to cleansing the blood and producing bile, we also see that they're processing blood-borne nutrients. One of the nutrients that they process is glucose. And what they end up doing is they end up creating these long chains of glucose called glycogen. And glycogen is a form of stored glucose, and it's stored in the liver. Our skeletal muscles also make glycogen from excess glucose in the bloodstream, and so the skeletal muscles will also store glycogen. But in the liver, glycogen is stored so that it can be used to make glucose, or glucose can be freed or liberated from glycogen between meals. So that way our blood glucose stays relatively consistent even if it's been a long time since we've had a meal. Right? And as we talked about in the blood module, the hepatocytes also function in making plasma proteins like albumin. Right? So looking at some homeostatic imbalances of the liver, we see that hepatitis is a viral infection that impairs liver function, and cirrhosis of the liver is caused by chronic inflammation of the liver. Two causes of cirrhosis of the liver are chronic hepatitis, that is chronic viral infections of the liver that are going to that are going to slow down the hepatocyte's ability to work properly, or even heavy alcohol use will eventually cause inflammation of the liver and liver cirrhosis. And what happens, the consequences of this are that the liver becomes fatty and it also becomes fibrous. And so we see that it accumulates fibrous connective tissue when the hepatocytes die. And so it's unable to function at its typical capacity because of that. So that concludes our segment on the liver. We'll turn our attention briefly here to the gallbladder. The gallbladder is a small, thin-walled muscular sac, so its walls are lined with smooth muscle, and it's located on the ventral surface of the liver, typically on the right side, or at least towards the right side of the body. One of its primary functions is that it stores and concentrates the bile salts, and its second function is to release those bile salts by contraction. And so when the gallbladder contracts, it pushes those bile salts into the cystic duct, which will then drain into the bile duct, and the bile duct eventually will deposit those in the duodenum of the small intestine. Looking at some homeostatic imbalances of the gallbladder, we see that gallstones can be created, which are also called biliary calculi. Typically, the cause of gallstones is excessive cholesterol production, either by the body or excessive cholesterol intake through the diet, since bile, as you probably recall, is a cholesterol derivative, and so too much cholesterol can create gallstones within the gallbladder. And these tend to obstruct flow from the gallbladder, and it leads to a condition called obstructive jaundice, when bile salts tend to build up in the blood because they are being released into the blood but not being picked back up. And that's one of the signs of obstructive jaundice is typically a yellowing of the skin.